All right, guys, so let's look at 3A3 a little bit more closely. So 3A was dealing with genetics. And so we're going to look at 3A3, where it's going to actually talk about some chromosomal disorders that affect humans. Now, a disorder can only be passed on from one generation to the next if it occurs during meiosis, okay? It has to be in the gamete because remember, you get your genes from your egg or sperm that comes from your parent. So the disorder has to be in the gamete. So any disorders or any mutations that occur in the somatic cells while an organism is living cannot be passed on to its offspring. But if their mutation happens during meiosis in the gamete, it can be passed on. Now, the difference between a chromosomal disorder and a gene disorder, the chromosomal disorder has something to do with the whole chromosome, either in structure or number. So if there's going to be a change in the number of chromosomes, humans should have 46. So if there's a change in the number, it's going to be caused by non-disjunction, which means the chromosomes did not separate properly during either anaphase 1 or anaphase 2, which led to a gamete that had either too many or not enough chromosomes. And if that gamete was fertilized, then the organism that is born from that fertilization will have either too many or not enough chromosomes. The other type of chromosomal mutation that can happen is a change in the structure of the chromosome. Now, this is caused by issues during crossing over, which happens in prophase 1. Remember, in prophase 1, your homologous chromosomes, your non-sister chromatids, so a chromosome from mom and a chromosome from dad, should exchange genetic information, but they should be of the same type. So chromosome 1 should only exchange information with chromosome 1. Now, sometimes when this happens, that crossing over has an issue where it's incomplete or something goes wrong. If crossing over happens and it ends up with more than one copy of the gene, that's called a duplication. So the normal chromosome should have genes A, B, C, and D, but if you notice in this chromosome, it has 2 gene Bs. And then, so that's a duplication. Now, hand in hand with a duplication, if one of the chromatids ends up with a duplication, then the other one's going to have a deletion. So where this one has 2 copies of gene B, this one has no copy of gene B. So this will be an example of a duplication, and this is an example of a deletion. Now, another type of change in structure would be if crossing over happens and you get it, but it actually is inverted or the genes are in the wrong order. So it should be A, B, C, D, but instead it's A, C, B, D, which may not seem like it's a big deal, but the location of a gene on a chromosome is important because genes are activated or deactivated based on their loci or their location on that actual chromosome. Another thing that can happen, remember crossing over should happen between homologous chromosomes. Well, sometimes chromosomes will get side by side and form a tetrad and they are not homologous. So in this case, crossing over has happened between one chromosome that had genes A, B, C, and D and another chromosome that had genes W, X, Y, and Z. This is called a translocation. Now, this translocation is going to actually affect 2 chromosomes because the piece of information that was exchanged, they're going to end up on, the genes are going to end up on the wrong chromosome. Trans means to move location. So the genes actually move locations to a different chromosome. So again, these are changes in structure. So you can have a duplication where you have more than one copy of a gene on a chromosome. You can have a deletion where you're missing a gene from a chromosome. You can have an inversion where the genes are in the wrong location on the chromosome. They're there, they're just inverted in the wrong space. And you can also have translocations that involves 2 different chromosomes and genes ending up on the wrong chromosome altogether. Now, when we talk about the human disorders, human chromosomal disorders, the 3 we want to look at are Down syndrome, Turner syndrome, and Klinefelter syndrome. Down syndrome is trisomy 21, where there's 3 copies of chromosome 21. So instead of having 46 chromosomes, there's an extra chromosome. So this would show 47 chromosomes in total. If you look at a karyotype, which is a picture of chromosomes, you're going to see that there's going to be 3 chromosomes at position 21 instead of the 2 that you should see. Remember, a normal karyotype, there's going to be 2 of each type of chromosome. So 1 through 22 are your autosomes. These are non-gender determining chromosomes. And pair 23 are your sex chromosomes, where females will have XX and males will have XY. Now, Turner syndrome and Klinefelter syndrome actually affects the sex chromosome, so pair 23. Turner syndrome is a disorder where it's called monosomy X. So it's a female that only has one X chromosome. So instead of having the 46 chromosomes that she should have, she's actually only going to have 45 chromosomes because she's missing one of her sex chromosomes. So instead of having 46, she's going to have 45. Klinefelter syndrome is a male with an extra X chromosome. So a male with Klinefelter syndrome is going to be XXY. Instead of having 2 sex chromosomes, they're going to have 3. Remember, males are hemizygous. They should only have 1 X and 1 Y chromosome. All right, guys, that's it on those disorders and the chromosomal mutations. Toodles.